Hello dear students, let's have a little view of the structure of chloroplast. What is chloroplast and where actually the light and dark reactions will take place. Chloroplast structurally is a double membranous organelles. These are two membranes, the outer membrane, the inner membrane space, and the inner membrane. And in these membranes there is a matrix. This is a fluid present that is said to be stroma. It's called stroma. To all this we consider it's a stroma. And here in a chloroplast there are thylakoids present, that we say small thylakoids and large thylakoids present, and these thylakoids are placed one above the other to make the structure of grana and intragrana and etc. I'm not going to detail the structure of chloroplast as we have studied already in a cell chapter, but I'm going to explain the structure of thylakoid in detail so that we can study the process of light reaction. This is one detailed structure of thylakoid, and I have already explained this structure, especially when I was dealing with the structure of chloroplast in chapter 4, the structure of cells. This is thylakoid. This is the membrane of thylakoid. See, this is a membrane of thylakoid, and in between membrane this is a cavity. So this cavity we consider this is lumen. This is a lumen, and this is actually a membrane. And in this membrane there are different kinds of pigments present that would help in the process of photosynthesis. In this thylakoid membrane, actually the pigment that we are studying, chlorophyll, is present, and these chlorophyll molecules are making systems. There can be photosystem II and there can be photosystem I. How actually these structures are present? Let's study them one by one. What is the arrangement of these structures? See, this is a photosystem I, which is actually made up of chlorophyll A that's present at center. And these are light harvesting complexes. They can be chlorophyll B and they can be chlorophyll carotenoids also. As these molecules are light harvesting complexes, they are called antenna complexes. And antenna complexes absorb light and they transfer the photon to a central chlorophyll A molecule, which finally is excited and emits electrons to pheophyton, which is the primary acceptor of photosystem II. Thus, this is a structure which is called photosystem II. And photosystem II absorbs light of 680 nanometers. Therefore, we say it is said to be photosystem II. And when electrons emit from primary acceptor, these electrons are received by another protein present in the thylakoid membrane. And that protein is called plastoquinin. And plastoquinin finally transfers these electrons to a complex that consists of cytochromes, actually two cytochromes, cytochrome complex B6 and cytochrome complex F. And electrons will move from plastoquinin to cytochrome complex. Actually, the cytochrome complex plays an important role, not only receiving electrons, but it pumps protons from its stroma also, and these protons will reach to a lumen. Therefore, we can say these cytochrome complexes are playing an important role in establishing a pump that is called proton pump. And what actually will happen when these electrons move from cytochrome complexes? These electrons will finally be received by another protein, copper-containing protein called plastocyanin. And this plastocyanin would finally settle these electrons to another system, which is called photosystem I. And that absorbs light of 700 nanometers. That was studied first and found first, therefore it's called photosystem I, and this is called photosystem II. What actually photosystem I? It also consists of antenna complexes and central chlorophyll A molecule and primary acceptors and everything. These are molecules, and these molecules are called chlorophyll B or carotenoids molecules. They are called antenna complexes. They will receive light and then transfer to a one reaction center, this chlorophyll A, that would finally excite. And when it excites, it emits electrons, and these electrons are received by primary acceptors. And from this primary acceptors, electrons are finally received by another protein that is said to be iron-containing protein called ferredoxin. And when this ferredoxin will receive electron, there are two ways to transfer these electrons. Either these electrons are transferred to NADP, and with the help of reductase enzyme, NADP is reduced into NADPH. As it takes electrons from ferredoxin and it takes protons from stroma, NADP is reduced into NADPH. And finally, these electrons are settled in NADPH, which is actually electron carrier, which would finally release energy for the synthesis of glucose. Therefore, this is a simple arrangement of the structure of different protein complexes, cytochrome complexes in the structure of thylakoid membrane. Sometimes when production of NADPH is raised or increased, plants switch to another mechanism where they start synthesizing ATP more faster, and then from photosystem 1, electrons that are emitted by a chlorophyll molecule. After ferredoxin, these electrons are then transferred to cytochrome complex. And when electrons are transferred to cytochrome complex, from cytochrome complexes, electrons reach back to a plastocyanin and they reach back to a SAM system where from they were emitted. So in this structure, you must be understanding, before starting the process, you must know how actually is the flow of electrons. See here, when electrons move from photosystem II, so they actually make a flow that is said to be a non-cyclic flow, where electrons are coming from photosystem II and they are reaching to a photosystem I. When electrons do not come in a same system, we say the flow is called non-cyclic flow. Here electrons will move from photosystem II and they will emit from chlorophyll A, then to a primary acceptor, and then to a plastocyanin, and then to cytochrome complexes, and then to PC. And finally, these electrons are received by a chlorophyll molecule which is having a hole here, as it is excited and its electrons are emitted, and finally when electrons reach here, this chlorophyll molecule is again stable. So this is a flow of electrons from photosystem II to photosystem I. Is said to be non-cyclic flow of electrons. During this flow, actually the protons are pumped inside lumen, and these concentration of protons keeps increasing in a lumen, which finally would boost up in a process of formation of ATP that we are going to study later. But the electron that comes from photosystem II and that reaches to a photosystem I and it emits again, and that can be received by ferredoxin, and same electron can reduce to NADP into NADPH. Resultantly, this non-cyclic flow of electrons can not only synthesize ATP, but it can also reduce NADP into NADPH. Whereas there is another flow of electron that can be from photosystem I, and electrons are received by primary acceptor and then by ferredoxins, and electrons from ferredoxins enter to cytochrome complexes, and from PC they reach back to a SAM system. When the electrons are coming out of one system and they are returning, they are cycling back in a SAM system, they are making a flow that is called cyclic flow, and that would also power to a proton pump, and that would also help in a synthesis of ATP. So by this process, by these two processes, either by non-cyclic process or by cyclic processes, actually protons are continuously pumped inside the lumen of thylakoid, and from this lumen, where protons are in a higher gradient, they would move from higher concentration to a lower concentration, from lumen inside stroma. They'll move inside stroma. And the study here another structure that's present in thylakoid complex, thylakoid membrane. This structure is called ATPase complex that works with the help of ATP synthase enzyme. And when protons from higher concentration, the osmosis takes place of these chemicals, these protons move from higher concentration to lower concentration in a stroma. As these protons move out of this complex, they provide energy which is stored in a phosphate bond, and synthesis of ATP takes place. And to this synthesis of ATP, we actually say this is actually ATP which is formed during light reaction, and that is said to be chemoosmotic phosphorylation. And to this we consider the chemoosmotic phosphorylation, as it is taking place because of light. Therefore, we say it's called chemoosmotic photophosphorylation. and we are going to study this in a process of detail. If this is a non-cyclic flow of electron and it forms ATP, you would say it's said to be non-cyclic chemoosmotic photophosphorylation. And if there is a flow of electron which is cyclic and it's providing ATP, that would be considered as a cyclic chemoosmotic photophosphorylations. And to this we are going to study in a detail later. There is another important complex also present, which is also the important requirement for photosynthesis, that's called water. With a photosystem II, there is a system present, there is a complex present. To that we say this is said to be water splitting complex. What actually this water splitting complex is, this complex breaks water, and to that we consider photolysis. And when water breaks in presence of light, it releases electrons and it releases protons. And these electrons and protons are released in a lumen, and then they will have to move to different waves. Electrons of the water reach to a reaction center of chlorophyll A, where it's excited, it has moved its electron out and it has formed holes here, and these two holes are finally filled by the electrons that are coming from water. And similarly, and this system will become stable. Thus water will keep breaking and it will keep donating electrons to a photosystem II and would keep stabling it, but it will again absorb energy and electrons will emit, and the process will keep going on. Whereas the protons that are coming in a lumen, these protons are protons are present in a lumen that are pumped by a proton pump. Whereas water is also providing protons, they would also be accumulated here in a lumen. And all these protons will move out of this complex and they will finally help in a synthesis of ATP. Therefore, water breaks by water splitting complex, and water splitting complex actually provides electrons to photosystem II and protons that help in a chemoosmotic synthesis of ATP. This is just the structure of a thylakoid, where you must be knowing this is called photosystem II, this is called photosystem I, this is called plastoquinone, these are called cytochrome complexes, this is plastocyanin, and with a photosystem I, there is a primary acceptor present that gives electrons to a protein that is called pyridoxine, and that gives electrons and reduces NADP into NADPH, which we are going to study in the process of photosynthesis. And this is a structure of a stroma. And in this stroma, when NADPH and ATP all are released here in a stroma, another reaction will take place, that is said to be light independent reaction or dark reaction, where carbon dioxide gonna be fixed into a triose, that is to release to synthesize glucose molecule, and this process is said to be a dark reaction that would finally complete the process of photosynthesis that we are going to study in a detail later when I'll deal with the dark reaction. This is the structure, general structure of a chloroplast, thylakoid and stroma and water splitting complex. This is general, and now I'm going to start the light reaction. Please subscribe my channel and share it with your friends also, and like and comment my videos, and let's learn together.